Welcome dear viewers to the video on topic of selecting the excipients for generic formulation development. The generic formulations are required to be safe, effective and with the required quality as like the reference formulation, innovator formulation or the RLD. The generic formulations are required to be equivalent in terms of bioequivalence, pharmacokinetics and the quality. The quality includes not only the bioequivalence but also the quality attributes related to the safety and efficacy. Whenever you are going to do formulation research, formulation development for the generic formulations, that time you will have reference formulation for comparison and for doing the bioequivalence study against it. So generic formulation is developed considering the reference formulation, and that's why whenever you are starting the development of generic formulations, always you should understand your reference formulation. I have made some videos on selecting the manufacturing process or packaging systems for the generic formulations, and this video is for the selection of excipients, selection of the raw materials for the generic formulation development. The generic formulations have different challenges like challenges of the patents or intellectual property challenges, which come in the category of legal challenges, then manufacturing challenges, stability challenges and the bioequivalence challenges. And you should consider all these challenges when you are selecting the excipients. Selecting excipients for generic formulation development requires the balance for regulatory expectations, bioequivalence, manufacturability, stability, and the intellectual property constraints. For example, if some excipients are patented, then if you are developing a formulation and if you use the patented excipients, then your formulation will be infringing. So that is required to be considered. Now we will start the video with more information. First is understand the reference listed drug. It is called as RLD. It is also called as reference formulation, comparator formulation or simply as a innovator formulation in line to which you are developing the generic formulation. So for selecting the excipients, review the RLD label, then check the inactive ingredient database, that is IID database. I have considered ANDA generic formulation presently, but this knowledge can be applied to all the formulations. Whether you are developing it for some other markets like Europe market, Brazil market, Canada market, so the knowledge will be same, the principles will be same. Review the RLD label and inactive ingredient database. Extract all the excipients at their quantities per dosage units if publicly available. So you can check with the patents, you can go through the RLD, you can go through the IID limits, you can go through the literature and you can get the quantities, because you don't know how much quantity of each excipient is used in the reference formulation. So for that you need to do some studies. Then check the US FDA IID or EMA databases for the maximum allowable levels. Understand your reference formulation before starting the development. Then second is analyze the RLD physically. So if you have RLD available, study it properly, disintegrate and identify the functional excipients. For example, if you are working onto a capsule formulation, open the capsule and weigh the contents so that you can get the idea of capsule size, you can get the idea of capsule fill weight. Or if you are working on a tablet formulation, check it, understand it, do some reverse engineering, do some QC testing, do some quality tests to understand the excipients. The common excipients for the oral formulations are like for tablets and capsule, these are binder, disintegrant, lubricant, diluents, film coating. Then check the ODT, ER, or film coating features requiring specific excipient classes. So if your reference formulation is film coated, then certainly you are going to do film coating. If it is delayed release, then your formulation will be delayed release, because you have to match the bioequivalence, quality and safety. So understand these things. Then define the formulation Q1, Q2. Q1 is qualitative sameness, Q2 is quantitative sameness, and Q3 is physicochemical sameness. So always I suggest you to go for this. Regulatory pathways dictate the excipient selection. Q1 qualitative sameness. Your excipient list must match the RLD excipient list. For example, if you are working on a tablet formulation, the RLD contains lactose, magnesium stearate, microcrystalline cellulose, povidone, cross carbamylose sodium, and HPMC based film coating. So your list of generic formulation should match this. Then Q2, that is quantitative sameness. Each excipient level should be within the variation range of 10% of the RLD amount, so that you can be in line to the reference formulation composition. Then Q3, it is physicochemical sameness. For topical complex products, match the microstructure. But I also suggest to get in-depth understanding about the RLD composition, because it will give you good idea, good understanding, and it will give you the confidence that your generic formulation will be in line, and that's why will be similar to the reference formulation, because your ultimate aim is to develop such a formulation which can be a replacement for your reference formulation. That's why try to match all these things. So if you have closer Q1, Q2, then there are higher chances that your bioequivalence will pass, your manufacturing process will be easy, you will have stability, and it will also reduce the justifications for the FDA queries or regulatory authority queries. Then consider API properties. So you should consider the solubility, you should consider the BCS class, whether the API is insoluble, or whether you are going to use some excipients to improve the solubility or not. So understand the API properties. API solubility and BCS class. So BCS class 1 and 3 are highly soluble, BCS class 2 and 4 are less soluble. So for 1 and 3, excipient should not alter the solubility or GI transit significantly, because it will affect the bioequivalence. Then class 2 and 4, excipient may need to improve dispersion, dissolution or weighting, because it is not like that you will use the same API like innovator. You will have different particle size, different uh, micronized behavior of the formulation. You may have different static properties, you may have different form, particle size, or sometimes you may have different salt form. So depending on that, understand the API. Also, I suggest you to match the API properties. If form 1 is used by the innovator, try to use that form, or directly use that form if you are not working onto a non-infringing type of formulation. So try to get the information about API properties. Then API sensitivities. So if API is hygroscopic, if it is photolabile, or if it is uh, labile to the compressive compression, then understand that. If it is hygroscopic, avoid the moisture retaining excipients like sorbitol, crosspovidone, povidone. If it is photolabile, you need to use film coating with TiO2. Compressibility issues are there, then use macrocrystalline cellulose or use the fillers which are uh, good for the direct compression process. Then API dose load. So how much is the tablet weight, capsule fill weight, or any other formulation weight, and how much is the dose load or API load in the formulation. So high dose if 100 more than 100 mg, low dose if it is less than 10 mg. So roughly I am saying you. So high dose filler choice is less critical and flow aids often needed. Low dose require excellent content uniformity, use high surface area dilutes and the glidants. So depending on your formulation, depending on the API load, depending on your manufacturing process, you can check the excipients, you can use the excipients and their quantities. Then choose excipients based on functional role. So select the excipients based on their roles like fillers, diluents, binder, disintegrant, lubricant, glidant, coating excipients, and many others. Lactose anhydrous, lactose monohydrate has the good compressibility along with MCC. It will give good binding. Mannitol is non-hygroscopic. It is more stable and it is good for disintegration, ODT type of formulations. So try to match the RLD in the excipients. Then binders, different binders can be used like PVPK30 is there, povidone is there, HPMC, HPC, starch paste you can use. So try to meet the binder similar to the reference formulation because binder level controls the hardness, friability and dissolution. Disintegration is required for the immediate release type of formulations, whether you are working on to tablets or capsules. So go for selection of the excipients like disintegrants. Try to use or match the RLD. Lubricants, magnesium stearate is there, sodium stearyl fumarate is there. So ensure the compatibility. One of the most important thing is meeting the compatibility. Then glidants like talc, colloidal silicon dioxide can be used. Coating excipients can be used based on the formulation design to meet the release profile like immediate release, delayed release or enteric release or enteric coating. HPMC, PEG, titanium dioxide alternatives along with different plasticizers can be used. Enteric polymers like HPMC, phthalate, udrazit and PVP can be used. So always select the excipients based on your RLD and the excipients available in the reference formulation. Then regulatory and safety requirements. So you need to meet the IID requirements. Pediatric formulations are manufactured with less quantity of ethanol or avoid ethanol, propylene glycol, sorbitol in more amounts. Then avoid allergens present in the excipients like lactose, gluten risk materials. Then check out any restrictions of some excipients like in past time titanium dioxide was restricted in some of the EU formulations. I hope now it is not there. You can use this, but always I am giving you just an example to check whether is there any restriction on some excipients. Then manufacturing process considerations. So based on your manufacturing process, you need to choose the or select the excipients. DC process requires good flow, less segregation, better mixing. So MCC spray dried materials, silicified MCC can help you. Then the materials which have consistent particle size, density and low moisture can be selected for the moisture sensitive APIs. Then for wet granulation, suitable for poor flow and segregation prone blends. So binders and fillers must be granulation grade. Hot melt extrusion modified release based on the technology, based on the dissolution profile required and based on the formulation design, you can select the excipients. So for hot melt extrusion, the thermally stable polymers and plasticizers can be used and required to be used, and these should have the compatibility with the API. Then IP assessment. So patent related issues may be there. Innovator may have filed some patents with some excipient quantities or levels, so that you should understand. If innovator has claimed that you cannot use mannitol as a diluent, then you need to select other diluent. Then check patents for RLD formulation and process excipients. Avoid unique property blends if patented for that use. Use GRAS or widely acceptable excipients to reduce the legal risk. Then conduct compatibility and stability studies. So compatibility is required to be performed to select the grades, select the quantities, select the ratios, select the excipients, and compatibility will give you the information with which you can understand the which excipients are degrading the API, which excipients are stabilizing the API, and really in which ratio. So depending on the API sensitivity, you can go for the drug excipient compatibility and perform the stability studies for the batches manufactured with different processes, different excipients. Excipient API compatibility can be done with TGA, DSC, FTIR, XRD, or you can check the impurities with HPLC method. Then accelerated stress studies can be done, stability indicating test can be done. Also check whether there is any incompatibility like Maillard reaction, acid-base interactions, water uptake issues. Always try to avoid hygroscopic excipients. So like that, you can select the excipients. Then develop the prototype and optimize it. If you have the Q1, Q2 data, then start with the Q1, Q2 matching prototype. Evaluate the properties like flow, compression, reliability, disintegration, dissolution, and always have multimedia dissolution to understand the effect of excipients. Adjust levels while studying the Q2 limits. Use the statistical softwares like DOE, design of experiments for the robustness testing, and load the batches on stability to understand the best composition. Confirm bioequivalence. So excipient should support matching dissolution profile with F2 similarity, multimedia dissolution, and should give you the stable in vivo performance, stable formulation. No excipient mediated variability should be there. Like mannitol sorbitol can accelerate the drug transit. Sodium lauryl sulfate can increase the solubility. 280 can increase the solubility. So like that, the functional excipient should be selected. So in principle, some points I have enlisted here for selection of the excipients for the generic formulation development, like match the RLD excipients wherever possible, match the Q1, Q2, do some reverse engineering and understand the quantity. Then ensure compatibility with API, physicochemical and biopharmaceutical needs. Stay within the regulatory IID limits. Consider manufacturing feasibility and intellectual property related claims like patent claims. Validate the choices through stability and bioequivalence studies. So you can do multiple activities at a time in parallel so that you can save your development time. I think this video got little bit lengthy, but it was necessary to cover the points, and I hope this video will give you good idea, good understanding how to select the excipients. See, as we are working professionals, always we should try to decrease our workload and improve our chances of success, so that these type of videos, informative understanding can reduce your time and can improve your output, can give you more success in less time. So understand the principles of selection of the excipients, and I suggest you to go through my videos. It will be very helpful to you to understand many of the topics. Thank you for watching the video.